to start off, I'm going to introduce Dr. Robin Hsiung, who is a behavioral neurologist with a extensive experience in clinical trials work. He has run many large trials with our clinic. He has a strong background in genetics, and he will speak with us this morning about the topic that Dr. Nygaard introduced, the amyloid in the brain and what's new with amyloid-lowering therapies. Thank you, Robin.
So thank you, Dean, and thank you, Hsiung, for introducing and organizing this forum so that we can still continue with our forum and provide information to everyone in our clinic and their families. So I'm going to share my screen. And now I don't see the rest of the screen, but are you able to see my screen? Anyway, I don't know, Dean.
Yes, Robin, it looks good. Looks good,
looks
Robin.
good. Okay, then I'm going to go on because there are lots of information in the next 15 minutes. So with Hsiung's introduction, we already talked about amyloid as an important part of the Alzheimer's pathology. And why do we want to lower amyloid? So there are different theories, but most importantly, amyloid is part of the pathology, which you'll also hear a little bit more about. So because of the plaques, amyloid plaques, we know that excessive amyloid is part of the Alzheimer's disease. So it's probably not very good for the brain. And all the genetics related to early onset Alzheimer's disease leads to excessive production of amyloid. So we know that all these genes leads to excessive amyloid and they lead to young onset Alzheimer's disease. So this is also evidence that excessive amyloid is probably not good for the brain. And now there are more laboratory studies studying the products of amyloid beta, a particular size of the amyloid, which we call amyloid beta 42, is actually toxic to the brain cells. And then when we try to suppress this in animal models of Alzheimer's disease, we're able to see arrest of disease progression. So this is a mouse brain with excessive amyloid production with the gene produced, put into the mouse. And if you stop that or by an immunization method, these mouse don't have amyloid in their brain anymore and they do better. So based on these findings, one of the major treatment is to try to lower amyloid. There's still some theory against this amyloid, and I don't have time to go into it, but obviously tau is another important pathology in the brain, and this doesn't really quite address tau, although there's some studies suggesting that excessive amyloid will lead to tau accumulation as well. But I won't go into the other mechanisms. There's still a lot of other things that are being investigated, but today I'll just focus on the amyloid. So how do we lower amyloid? Based on the genetics, we know that amyloid is a part of this APP protein, amyloid precursor protein, and is an important protein in the membrane of brain cells. And it's processed in different ways. If you cut it in the middle, this red part will be broken up and it won't be a problem. But in patients with Alzheimer's, it's cleaved just on top and in the bottom of this red chain. And when it's released into the bloodstream or in other parts of the brain, they tend to accumulate. And when it accumulates into a certain size, it's actually also toxic to the brain. So that's why the theory of lowering amyloid may be beneficial to Alzheimer's disease. And the two ways to inhibit it, one is to use an inhibitor for the enzyme that cuts this protein up. And another way is to use a monoclonal antibody to bind it and remove it from the other parts of the body. So hopefully this can be leaving the body. So UBC has been involved in clinical trials ever since the development of treatment for Alzheimer's. But in the last 5 years, things have been mostly very disappointing, especially with all the base inhibitor, the enzyme that cleaves amyloid. They were all negative, and some of them actually get worse while on treatment. And this suggests that this enzyme probably has other functions that we don't know about that is important to the body. And if you just cut it off, it's not that useful. And then in the more recent years, we were looking at monoclonal antibodies, which is a very effective way of removing amyloid, but not all of them work the same. Some of them were not very helpful in the early studies. And then the last three were actually now showing some positive results. And I'll go into these two studies particularly because the results were released over the last year and they were a little bit controversial. So I'll spend a little bit more time talking about these two studies particularly that came out. So now this aducanumab, Hsiung already showed you the slide earlier in 2006 when it first came out with the preliminary study. It was very dramatic because the accumulation of amyloid in the brain was actually mostly reversed. So based on those findings, we were able to move on to a larger scale clinical trial, which is the Engage and Emerge study, which happened between 2016 and 2019. They moved from a phase 1 to phase 3, and the different phases are different size of a population. And I think they were so optimistic that they skipped the phase 2 and moved on to phase 3. And when you have two phase 3 being positive, that will allow the company to actually seek for approval of the drug. So this was a little controversial because it was on the news quite a bit in 2019 because while we were going through the study, it was quite optimistic that people were doing well. But when they analyzed the midpoint, which is at the end of December of 2018, they used something called a futility analysis. And they look at the first half of the people. So the first 50%, approximately 50% of the people who got into the study, they started analyzing the data. And then in March of 2019, they released the data and told us that based on the futility analysis, they said that it was negative. So it was very disappointing because it was very promising in the phase 1, but we have to stop people. But we don't stop people immediately. So there was a phase cutoff. So over the next several months, we actually stopped some of our participants in the study, but we completed collection of the data. And that ended sometime towards May of 2019. And then surprisingly, towards the end of October, early November, the company released a different set of results suggesting that it was actually positive. And what they did is to look at the further number of people who actually completed the study towards March and to May of 2019. And then they also included everyone who had some sort of data, and that is called the intention to treat. So everyone who actually was in the study at some point and have data collected, they were able to include them in an intention to treat analysis. And then there are a partial number of people who completed the PET part, which is the amyloid scan, and those were released. So in that study, when they look at the Emerge result, Emerge is one of the parallel study, they found that there's actually significant improvement in both the intention to treat and the opportunity to evaluate. And in those studies, they found the CDR, which is a clinical measure of how people are doing in terms of their function. They find improvements. More negative means more improvements from the baseline, or actually less less progression, sorry, not improvement, but there's less progression in the people who got a high dose treatment. And that was true in the intention to treat population as well, the opportunity to analyze. And there are some secondary endpoints, which are the cognitive outcomes. Mini mental status is a common measure that we use in the clinic. And the ADAS-Cog are more comprehensive measures that we use in clinical trials. But in this particular study, the emerged people were improving, or at least not progressing as much in the high dose treatment. So based on this, they actually called it a positive study. And that was released towards the end of October of 2019. However, there was another parallel study, the ENGAGE study, and they did not see the significant improvements compared to the other study. And we don't really have a good explanation of why they are not improving. And if you look at the particular measure of CDR, they are actually doing a little bit worse than the other group. Although in the other cognitive measures, they are almost the same. And in this particular group, the placebo are also not progressing as much as the other group. And that's why there's no significant difference in this particular trial. And then they look at the amyloid PET scan, and the red group are the placebo group, so there's no change in the amyloid accumulation. But in both the EMERGE and the ENGAGE study, people have significantly reduced amyloid in the brain after about 2 years of treatment. So the low dose reduced it about by 15%, and the high dose reduced the amyloid by about 30%. So again, this confirms the finding in the preliminary studies based on the amyloid PET scan. So these people are actually removing amyloid based on the PET scan measure. And interestingly, you hear a little bit more about spinal fluid, cerebrospinal fluid biomarkers, but in tau and phosphorylated tau and total tau, which is more of a measure of brain cells degeneration, there's also a signal of reduced CSF tau production in both studies. Although again, in the EMERGE, there is a clear dose response, so the higher dose was working better. But in the ENGAGE study, there's some signal, but not as strong as the EMERGE study. But the number of people who actually got completed spinal fluid are not very high. So there were almost 1,000 people in the clinical trial, but there were only a small number, 20-something people per group who got the spinal fluid analysis completed. So it's only a small subset of the people who actually completed the trial. But based on this finding, Biogen, this company that is currently doing the trial, has issued a news release that they are going to try to apply for approval. We'll see what happens. From my point of view, this data is not strong enough to approve the study, but they are encouraging. And we are going to recall all the participants of our clinical trial to come back for an open label extension. So those people who were in our study will be called back as soon as we get approval to complete the study for another 2 years. And in the open label, everyone will get the real treatment. There won't be any placebo. So related to amyloid treatment, there's another study that is called the Autosomal Dominant Inherited Alzheimer's Network. It's called the DIAN network, and there's a treatment unit. In this first phase of the study, we are doing a 3-arm study to compare a placebo to 2 other amyloid-lowering agents, gantenerumab and solanezumab. And these are for people who have a family history of known genetic cause of Alzheimer's. You may hear a little bit more about familiar Alzheimer's disease from our geneticists later on, so I won't go into the details. But these are family members with known mutation that causes early Alzheimer's disease, and they can be in the at-risk phase, so they just carry the gene, but they're actually normal, and they can be in the very mild dementia range as well. And they also get MRI and PET scans and lumbar puncture. It's a very difficult study. It was conceived at the end in 2011, so almost 10 years ago now. And in the first phase, just trying to get people in, it took about 3 years just to have the cognitive endpoint or in the phase 2 biomarker studies. And UBC was involved in the phase 3 when we were actually enrolling more patients to treat them in one of the arms and follow them through. But it was back in 2016 and 2017, the gantenerumab and then solanezumab have preliminary studies from other clinical trials suggesting that the dose that they used was safe, but actually not enough. So halfway into the study, these doses were increased. So the gantenerumab was escalated about 5 times from the baseline, and the solanezumab dose was increased about 4 times compared to the baseline. So
2 minutes, Robin.
This was a little bit controversial. Am I running out of time? Anyway,
yes, you are.
Okay, so I'm going to skip to so the summary is that the clinical endpoint are not really different. So in the placebo group and the treatment group, there's really no significant difference in the worsening of the clinical measure. But again, intriguingly, the biomarkers are quite significant, especially for the gantenerumab. So in the gantenerumab, in this particular study, there's significant reduction in the PET measure of amyloid accumulation in the treatment group. And there's also significant improvement in the spinal fluid analysis. So there's reduction of amyloid in the spinal fluid, and there's also reduction in tau, which is a measure of brain cell death. So based on these biomarker studies, I'm going to skip the details. These are encouraging results. So the aducanumab shows some clinical improvements in partial data in part of the clinical trial. But it is a bit controversial because the clinical trial was terminated early. So they are based on their analysis with incomplete data. And there are some differences between 2 parallel studies. The findings are more significant in the emerge, but not in the engage. But they are exactly the same study, and why are they not the same? So the detailed analysis are still pending, but it is intriguing, so it deserves further attention. And in the Diane study, neither the gantenerumab or the solanezumab showed any clinical improvements, but the gantenerumab shows a decreased amyloid accumulation as well as improvement in the spinal fluid measure, which is a measure of disease progression potentially. I didn't have time to go into the adverse effects, but both of these amyloid-lowering agents causes something called amyloid-related imaging abnormalities, which is basically fluid accumulation measured by the MRI brain. But most of these effects are reversible, so people didn't have any persistent worsening, and if you stop the treatment, people reverse, so they got better. So there are a number of promising studies in the pipeline, and both of these studies are being continued, and hopefully we'll have more encouraging and positive results when we have longer studies. Thank you very much. And sorry I went over time.